Having looked at the main replication enzymes involved in DNA synthesis, let's consider more of the proteins and enzymes that participate in this highly sophisticated process. Since there are multiple participants in this process of replication, a complex known as the replisome forms at the replication fork. This complex includes DNA polymerase, the RNA primer required for synthesis, the primase enzyme that generates the primer, and the helicase enzyme needed to separate the DNA strands. A challenge to the process of replication has to do with relaxing the supercoiling tension on the molecule so as to separate the strands. Also, exposed regions of single-stranded DNA must be protected from nuclease digestion. Since prokaryotic DNA is negatively supercoiled, separation of the two strands results in positive supercoils ahead of the replication fork, as illustrated below. In a previous lesson, we learned of topoisomerases that alter DNA supercoiling. DNA gyrase is a class II topoisomerase that introduces negative supercoils with concomitant ATP hydrolysis. The action of placing negative supercoils ahead of the replication fork serves to reverse the effects of the positive supercoils that form as a result of strand separation. This also ensures that the newly synthesized DNA adopts the correct supercoiled state. In the figure below, you can see a depiction of DNA gyrase as the pink disc ahead of the replication fork. Now notice the blue-green oval shapes that immediately follow the gyrase enzyme. These represent the action of the helicase enzyme that separates the strands and the primase enzyme that generates the RNA primer. The single strands of DNA are protected against nuclease digestion by being coated with multiple copies of single-strand binding protein, pictured as the bright green spheres. As you can see, the process of DNA replication is carefully choreographed between multiple players with various functions carried out in a very specific way.